Thank you, Dr. Porter. What a joy to be with this group today on such a beautiful, I'm going to say spring day. The spring is just about here. And also, what a joy to have Dr. Blood here. What a great colleague Dr. Blood was. You folks that have heard my lectures on acid rain, Dr. Blood was doing work on acid rain in the 80s here on the high probability, some of the only work ever done in South Carolina. Went on to a stellar career at NSF, where she headed the NEON program, the National Environmental Observing Network, manager of that, just a wonderful career. Great colleague and friend. She was with the Coca-Cola Foundation. She just had quite a stellar career. Just what a joy to see you here today, Dr. Blood. And boy, what a fun time. Well, today I'm going to talk to you in our field seminar. And by the way, what does field, P-H-I-E-L-D, stand for? Well, public health. In environmental learning and development. So that's what we're going to hopefully come up with something to capture the whole in that. Today, I'm going to talk about, since we're at the coast, ocean and human health. A One Health concept whose time is coming. I could not even start or even think about doing that without Dr. Porter at my side. And Dwayne and I have been doing work together way longer than I care to admit sometimes. So a number of colleagues at our Center for Oceans, we're one of 4 national centers. Let me go back. These are the other national centers: Widsall, Bowling Green, the Great Lakes Center, Florida Gulf Coast, Caribbean Center, our Center for Oceans and Human Health and Climate Change Interaction. All the other centers are funded by both NSF and NIH. We're the only center that's only funded by NIH. So because of our human health focus. Okay, these are some of our colleagues. Dr. Paul Sandifer, a host of folks at USC, including this young lady right here. And Dr. Weinstein at the Citadel, Brian Brooks and Thad Scott Bale, Shuo Zhao and Yingqing Wang Yet. Right. So we talked about One Health in class this week, right? And that really recognizes the interconnection between our health and the environment. And nowhere is that more clear than at the coast. And in fact, One Health is important because, as we said this week, 6 out of every 10 infectious diseases in people go back to animals. So, ocean health, we've been studying it for an awful long time. Dr. Porter and I spent most of our career early, and Dr. Blood, looking at the effects of man on the oceans. How are we screwing up the health of the ocean? Then one day, Paul Sandifer and the late Senator Fritz Holland got the idea that, well, you know, if the ocean is not in good health when we're living there, maybe that might not be good for us. So Senator Holland introduced the legislation for ocean and human health research. And so that really, we've been studying that. So we really flow that entire circle all the way from man to the ocean and ocean to man. So our mission at our center is to assess the effects of climate change and climate change interactions. I like to make this point. A lot of people are studying climate change. They're studying the effects of temperature, effects of salinity, sea level rise. We're looking at a much more in-depth level of interaction. And as we go into this talk today, I'm going to be talking about it. We're looking at 3 stressors: infectious microbes, harmful algae, and microplastics, and how they interplay and enhance each other in the environment. So our research is really focused on toxin production from harmful algae, infectious microbes and vibrios, and then microplastics. So these are all the cast of characters: Sean Norman, Brian Brooks at Baylor, John Perry in our chemistry department, Ron Chatterjee, University of California, Irvine now, and Dr. Porter who heads up our community engagement corps. So we have micro, freshwater, harmful algal blooms, chemistry related to that, toxicology, the human health effects of these toxins and microbes. And the model we use are what we call humanized mice. They actually have human organ systems in the mice. So when we talk about effects in mice, we don't have to make a leap from mice to men. We're already dealing with human organs, which is we're one of the very few centers in the country that have this kind of humanized mice. So why should we do this? Well, we're currently spending over $1 billion a year in healthcare costs related to marine pathogens, foodborne illnesses, harmful algal blooms right now. And if we add in the freshwater component, we're spending almost $2.9 billion in healthcare costs. And those are for the reported cases of illness. That does not go with all the unreported cases. So for some of the biblios that we talked about, for every case that's reported, we have 30 cases that go unreported. So these are just scratching the surface in terms of estimated healthcare costs. Why do we focus on the coast? 17% of the land area house almost 50% of the world's population. They house over 50% of the gross domestic product for industry produces things, because we ship things from the coast to have international commerce. So everybody is located on the coast. So that means we have 5 times more people per land area. With something like COVID, you think that might be a big factor that will crowd it together? You bet. And also, when we have that much 5 times more economic activity, that means air pollution problems, water pollution problems, always are worse at the coast than anywhere else you go. You know, Kansas is a lot different than Myrtle Beach in so many ways. Okay, so today we're going to talk about basically how these impacts from the coast come to play in terms of affecting infectious microbes, causing harmful algal blooms, and then also the proliferation of microplastic pollution. So when I like to talk about the effects of climate change, you cannot talk about it without mentioning the word urbanization. My stock talk for probably 15 years was that the interaction of climate change and urbanization, a recipe for disaster. That was my standard seminar wherever I went for 10 or 12 years. But oceans and human health have now become my friend. And it's because climate change really is the gasoline we're throwing on top of urbanization when it comes to environmental impacts. Urbanization, we pay paradise and put up a parking lot. And we change the hydrological cycle, we change the delivery of the water and the things in the water, the pollutants. We specifically affect the nitrogen and phosphorus cycle, and that leads to eutrophication. And that leads to toxic algae. 87% of our estuaries are highly eutrophic. That means they have too much nitrogen and phosphorus and not enough oxygen. If your body doesn't have enough oxygen, are you healthy? No. And neither are our coastal areas. So climate change, we alter a force cycle, the carbon cycle. So you actually think of the ecosystem. When we talk about the word ecosystem health, when I talk about your health, I'm even talking about your lungs, your heart, your kidney, your liver. So that's your reproductive system, whatever it may be. When we talk about the ecosystem, these are the systems we talk about. These are the organ systems that matter to the environment. And so in the same way we talk about human health, we have to talk about ecosystem health. And like I said in that little part, they're intimately related. So let's talk a little bit about carbon dioxide. We know that is increasing. Y'all have all probably seen this slide of CO2 levels from Mauna Loa in Hawaii. These are the levels in the atmosphere. These are the levels in seawater because CO2 absorbs into seawater. And that is the declining pH in seawater because of the acidic acid, the carbonic acid form that lowers the pH and causes ocean acidification. Presently, CO2 levels somewhere around 420 parts per million. They're continually rising, and it increased by 25% since the start of the Industrial Revolution, 12% since 1960. We estimate that's caused about a 1 degree Fahrenheit change in the 20th century, and by the end or middle of this century, a 1.5 to 4.5 degree Fahrenheit increase in global temperature. That's not extreme temperatures, much worse at poles than, for example, at the equator. So this is a kind of an interesting graph. This is temperature variability over time, 1880 to 2005. So when I talk to people about climate change, well, Geoff, there are natural things that can cause temperature change. Orbital wall, or what we call the Earth kind of doesn't always spin the same. And solar sunspot activity, volcanic activity. So you'll notice that if we just take those three factors, right up to about 1960, they track pretty good in explaining most of the variability in temperature. But notice, after then, something else is going on. Now, if we come down here and look at man-made factors, land use or urbanization, levels of ozone, aerosols, and greenhouse gases, notice they track perfectly in the models that predict increased temperature. And they particularly track where this does no longer, these natural factors no longer track. So the leading, greenhouse gases of CO2, about 55%. And that level, this is an old slide. I think that was the CO2 level about 2010. It's continually going up. CFCs, these are the chlorofluorocarbons from that air conditioning in this room. Every molecule of CFC, though, it absorbs 12,000 times more infrared radiation than a molecule of CO2. And they're increasing at 5% a year, so they're doubling every 20 years. And as it gets warmer, guess what? More places are installing air conditioners, so these are going up even quicker and more and faster. Methane, 15%. They're 20 times more potent than CO2 at absorbing infrared radiation or heat. And then nitric oxide, they're not increasing quite as much. While we have catalytic converters on our cars, thank goodness, and we reduce those emissions. Now, here's a slide that I like to show people. This is essentially looking at the level of carbon dioxide from the fossil record. Everybody always says, well, you don't go far enough back with that slide. So we're going back almost 600,000 years. And yes, CO2 goes up and down. But look at that trend. This is what's different. It's the rate of increase. And notice when that happens, when those two lines no longer, natural variability can no longer explain the change in temperature, that's when we cross this upper limit that's naturally occurring. And this is what is all that is happening that really leads to really showing you, and these are ice core data looking at the fossil record. So today, I'm going to talk with you about climate change and how they really directly affect the growth, survival, persistence, transmission, and virulence of disease-causing organisms, Vibrio bacteria, harmful algal blooms, and also a little bit about microplastic interactions with those. So you've already seen this slide and this slide, so I'm going to skip through. And sorry, I've been having trouble with this. For some reason, I'm going to have to get out of this. There we go. Okay. Oh, good. So Vibrio bacteria, and particularly this is Vibrio vulnificus and Vibrio, did I miss it?
No, it's okay. I'll do it.
So these are extreme lesions from Vibrio vulnificus. When you get this, you got about 2 days to do it. They can either cut your hand off or leg off, hopefully get the right antibody to save your life. If they choose the wrong antibody, you're pretty much toast. Rita Caldwell, colleague of Liz's at the NSF, for a number of years, director of NSF. Rita's shown over the last 10 years globally, Vibrio levels of infection have increased by 41%, and there has been a 31% increase in the amount of antibiotic resistance in those infections. In the US, we've gone from 8,000 estimated cases in 2004, a tenfold increase by 2019. And so this is Vibrio vulnificus. This is not Vibrio cholera, it's first cousin. And it causes 95% of all the deaths from seafood consumption. So let's talk a little bit about Vibrios. These are just some statistics. We now see cases all up and down the coast of South Carolina and North Carolina. We also see cases in the North Sea, the northeastern US, and we see them increasing higher and higher global latitudes. And so the Intergovernmental Panel has already made this estimate, which I gave you earlier. And this is just a heat map showing that change they predict over time. So this is a good example of what we see with Vibrio illness. In 2014, we had an outbreak in Alaska. This is normally an organism that occurs in the Gulf of Mexico. You had a 2,000-kilometer range extension up to the Gulf of Alaska. And for the first time in recorded history, the water temperature in that region went above 15 degrees C, according to the National Weather Service. And within a matter of weeks, we began to have Vibrio illness. And so it was clearly related to the increasing temperature. So why do we have more cases? We have more geographical areas, wider areas with higher temperatures, and we have a longer season for the Vibrios to grow. Somebody sent me an email this week, Geoff, do you think it's safe to eat oysters as hot as it's been lately? Because this is the weather we normally have in April at the end of the harvest season. Question. I told them, make sure you cook them. So them raw. Okay, so let me show you some statistics with oysters. That's kind of a complicated slide. These are decadal levels of cases of vulnificus or Vibrio from 1989 to 97, and this is per metric ton of oysters. So these are the percentage of days above 20 degrees C in the month of April and the month of November. So this is the next 10-year decadal rate, average from 98 to 2007. Notice the increase in the percentage of days where it's warmer, making that season longer. Particularly notice in November. And then look at the number of cases of illness, how much they have gone up in those 2 decades. Dramatic increases with increasing temperature. So again, it's space and time. That's what raises the number of cases of illness that we see in seafood. Now, Vibrio is a really interesting disease. If you see a blue line like Vibrio parahemolyticus, about 85% of all the cases are in oyster shellfish. If we look at Vibrio vulnificus, some cases in food, most of the cases are now wound infection. And according to Rita Caldwell, 30 years ago, we didn't hardly ever see a wound infection. It was rare. Today, with the increased virulence and temperature in antibiotic resistant Vibrios, Vibrio alveolyticus is almost exclusively a Vibrio that causes wound infection. And why does it have the name alveolyticus? because it's correlated with its presence with harmful agents. We're going to get to that intersection in just a minute. So you'll notice each of these vibrios that cause quote unquote vibriosis can have multiple ways in which they can make you sick. Now here's another interesting graph. In the 1970s, these are recorded percentage cases of illness. Only 10% of our illnesses were in molluscan shellfish. 90% of our illnesses were in fish, sigmatoxin. You'll notice though today, only about 25% of our cases are from fish, and the majority of our illnesses today are in oysters and also in shrimp. Again, those are vibrios as well, because the vibrios attach to the chitin in the shrimp, and that's a very unique. Caldwell was the first person to discover that in her research at the University of Maryland. Now, one of the things we have done is how do we protect people from these illnesses? Well, we can't monitor everywhere, but what we can do is develop forecast models like Dr. Porter has House My Beach, and he can tell people when they should not go swimming. Well, in the same way, we're developing Vibrio forecast models. And so we work with a guy called the late Paul Conrad, who took 5 different global climate change models and created this neural network model that would predict increased precipitation or decreased precipitation, depending upon whether it was a drought or flood related year. And then he would then, if it was a wet year, he could then take an exam prism model and predict increased river flow to the coast. If it was a dry year, he also had a sea level model that would predict saltwater intrusion further in our rivers upstream. And right here, this was all done right here in Minnehaha Bay, was our model watershed. So we trained the model over this time period and then forecasted out to the future. So this is the salinity change with no sea level rise, a 1 foot sea level rise, 2 and 3 foot sea level rise. These spikes in salinity actually tell us when we're going to have problems with our drinking water here in Georgetown, because it's going to be too salt. And so you can see as sea level rises going all the way out to 2068, we're going to have some problems with our drinking water, not only here in Georgetown, but all up and down the coast. Well, we can then take that model for temperature and salinity. It also predicts temperature. And this predicts spikes of Vibrio volnificus. And so this is with no sea level rise in 1 foot, 2 and 3 foot sea level rise. Now, our stations were all the way almost up to Pawleys Island up in Waccamaw River, that lives with Kay and Wayne. That's fresh water. But it's not going to be much longer, because we're driving the salt water upriver with sea level rise. And that is making growth conditions 230% increase in optimal growth conditions for vibrios. So this is how we warn first. NOAA actually runs a forecast for vibrios for the Chesapeake Bay. John Jacobs used to be at my NOAA lab in Oxford, Maryland. John runs that model and predicts the safety for folks coming in contact with the vibrios in Chesapeake Bay. Sea level rise is here to stay. And this is becoming a very common sight in everyday life along the coast. In fact, this model kind of shows it. Here's our average high tide today. If we get a little rise of sea level with king tide or a wind event, we get minor flooding. Moderate flooding will bring that water into our house. By 2030, this is where the average high tide is going to be. This is our minor flooding. Now our moderate flooding is occurring with greater regularity and frequency in our house. And then by 2045, here's the high tide, here's the minor flooding, and now the floods are in your house. What about all the pets that live in the yards where these houses are. They're getting wetted with water more and more frequently. That ownership is 70%. And that poop is all washing out right back into our waterways. That reporter I found with Steve Kelsey, for the first time in the state, the majority of closures in Merle's Inlet right up the coast. What were they doing? Pets. Everybody said, huh? So that's what we found. And the county is still working with problems up in Merle's Inlet in their dissertation today. So here's a little study we did out at Celala. They were having some problems with high levels of fecal or enterococcus through contact recreation and water standards. So Christina Eck, in her master's thesis, plotted the data, and the closures weren't occurring during the summer. They were all occurring in the month of September. That's kind of strange. What's going on in September? That's the time of the maximum thermal expansion of fever. Every year, that's what drives the hurricane season. That time of year, it raises the water level. So this is when we have the greatest amount of flooding from king tides. And if king tides wash up on the land, they were doing just like that little slide I showed you in the yard. They're wetting more and more of the beach. And if there are bacteria from animals on the beach, birds, dogs, then you're going to have more bacterial issues. In fact, when we look at what stage of the tide do the samples exceed the water quality standard, it's right before flood tide and right after flood tide. And in fact, at the time of flood tide, you'll notice that the other times, and the blue bars are the percentage of tides that fall in each of those categories in the samples we looked at. So the only samples where the violations exceed the occurrence of the number of samples these are percentages. Basically, you can see that they're disproportionately at flood tide. That also didn't work with our model because we used to see very high levels at low tide, but as we take a look, when we did the microbial source tracking of where all those bacteria are coming from, birds and dogs, this is 2 different years, birds and dogs. Now, what's unique about Edison Island in terms of dogs? Anybody know? It's the only beach in South Carolina you can take your dog out of the beach 24 hours a day. All other beaches restricted a certain time here, but Myrtle Beach, 6 p.m. to 6 a.m. You have to have them on a leash everywhere. The only other beach that they allow this, so when Christina did her work, she looked at when did they have sequences at these other beaches like Myrtle Beach? They don't have them in September. The only other place that did is Sullivan Island. And there you can have a permit, take your dog. It's almost just like Edison, only it's made convenient for the residents because the tourists don't go and get a permit. But the residents do. And it's the only other beach that had closures, most of the closures in the month of September. So it's clearly related to not properly cleaning up behind the fire. So here's a great example of climate change. And as we look up and down the East Coast in the 1970s in Charleston, we had 4 flood events a year. 2018, they had 70 events, I think, in 2018. Mayor Tecklenburg met with us at the Oceans 2018 meeting, which kicked off our summer going and told us, you heard he was having the folks in Denmark, or excuse me, the Netherlands come over and help figure out our flooding. You know what? This is, if you close the number of days in the year, about 70 days, a shot downtown in Charleston is going to do it. And if he doesn't fix the problem now, he will not have the tax base to fix it in a few more years. And so here's the real scary thing. If we look ahead, by 2030, we estimate 180 days a year of flooding, and by 2050, we estimate 240 of these events that will cause flooding. That's literally year round. And so that's 2030, 2045, and this is current. Flooding is coming, folks. It's coming quick. And coastal areas have to adapt. And that's why Mayor Tecklenburg is trying to go expeditiously as quick as he can. Because if he doesn't, he's going to lose his tax base for those businesses downtown. Be hard for the College of Charleston to hold class. Either that or we're going to have to invest in diamonds. Okay, so I want to take you back in time. Y'all are here, you heard this wonderful story about Bellwood and how she was she left a conservation gem to you in the state of South Carolina. And North Inlet is such a unique place. This is North Inlet. And in the mid-'80s, Liz and I, and Dwayne, we were all involved in the study. The first study to really comprehensively look at urbanization on an estuarine basis, not on an individual station, 30 stations in Murrells Inlet to the north and North Inlet to the south. Now, why North Inlet? North Inlet, you've already heard from Dr. Porter, is one of 29 National Western Research Reserve sites. So for you ethnic folks in the crowd, we always want to have controlled population. This is a controlled, unexposed, or least exposed environmental population, so that we can then compare things with a highly urbanized site like Murrells Inlet. So one of the things we looked at were the number of petroleum hydrocarbons in the sediments. So these are the 30 stations in North Inlet, maximum concentration about 3,000 or 300. And these are the stations in Murrells Inlet, maximum concentration almost 2,500, almost a 10-fold increase. So PAHs, petroleum hydrocarbons, are 10 times higher, and they spike at certain locations here at TP2 and GC3. Have any of you ever been to the beach at Garden City? This is the causeway going out to Garden City Beach. Every car going to the beach discharges their hydrocarbons on either side of the road. This site right here is one of our favorite sites. That's Grumpy Jack's. I'll let you figure out why. And so it's a marina. So the pollution sources are cars and boats. Dwayne counted back in 1988, how many boats per hour? 180 boats, Dwayne, I believe, traveling through the creek per hour. I was talking to Dwayne about this a couple of weeks. I've been to several thousand. Does that mean more people? And so if we plot the data, here's the data in Murrells Inlet, all the PAHs of where the roads are and the marinas. And then it drops off, and then when you get out to the mouth, there's not much there. It's about like North Inlet. Notice North Inlet, there is no spatial pattern. Where do the PAHs come from? This is the regional atmospheric depositional background in the sediment in North Inlet, and they're uniform. So this is why this long-term data is so important. So we kind of came up with this model. If you increase urbanization, you increase the levels of PAHs in sediment and oysters and crabs. And so we found, if we look at the levels of PAHs that we had, like at Grumpy Jack's, or the marina, or the roadways, this level right here, it really affected this little critter called a grass shrimp. That's a little shrimp, not important. It's only 55% of all the macrofauna, if you wanted that through these creeks annually. It's only 55% of all the macrofauna you'll catch in your net. A little important, it's the base of the food for all the 88% of the finfish that spawn offshore and come in here and make up our commercial and recreational fisheries. And without this organism, they don't survive. At a low concentration, notice there's not too much mortality. And then here's our control. So this is how we use what I tell you in class pickle jar ecology to figure out how significant things are. So we then predicted if we look in Myrtle's Inlet, what would the abundance of shrimp actually be if we did a population assessment? These are 2, 4, 6 sites. And here are our reference sites at North Inlet and another reference site in Charleston. Notice much lower populations. These are 2 sites, one out at Keel Island, one at another site at Waddell Island. Both impacted by agricultural pesticides, as probably remember we were doing this work. There was a fish kill here that wiped out 90% of all those little shrimp in 1 6-hour rating. It killed 99% of all the shrimp. And on an annual basis, it recovered, but that 1 6-hour exposure reduced the population. This is the same part of that same creek system. And you'll notice just that 1 6-hour exposure reduced the amount of fish you had. 1 6-hour rating. So our data really showed these PAHs were greatly, and the effects of the PAHs were much worse than the pesticide. There was one problem. We put caged animals, we documented all the shrimp died. When I put cages out here, it rained, nothing died. That's why I don't have a lot of care. So I began to try to figure out how do we address this? What's going on? So thank God, this is a gravid female shrimp. And they put out these little egg clutches twice a year. They have the first cohort, they'll be showing up with eggs April 15th in contact. And they'll spawn about half the number of young for the year. And then those young in April will mature, and there'll be this second wave of recruitment the second half of the year. And so if I look at North Inlet, 48% of the late season overwintering, and then, the overwintering of 52, and the second half of the year, that's that second cohort of shrimp. But then when I look in Myrtle's Inlet, if I go down here by the state park, it looks a lot like North Inlet. But notice, once I get up here to Drunken Jack's, the boat landing, and here's the Garden City Causeway where those high levels of PAHs were. We're missing the late season female. We're not getting that second wave of recruitment. Something's going on. Now, about this time, the literature began to show if you add ultraviolet light exposure, which happens with these PAHs, it makes them 10 times more toxic. Potential. So we did that in the lab, and then we factored all that in and ran our model, and we were able to show 97% of the effect on this reproduction was explained by the level of PAHs, temperature, and dissolved oxygen, and on the abundance of shrimp, we could explain 93% of the abundance just by runoff and PAHs. So we knew that PAHs were related. Then Dr. Porter came to the rescue. He measured the ultraviolet light levels in Myrtle's Inlet. And guess what? They were real high. Right there where that reproduction was altered. Now, folks, there's a risk assessment when you get stuff as close to a smoking gun as you're going to get. You don't, most of the time you get that bullet fragment and an effect. This is a smoking gun, the bullet fragment and the effect. So thank God for Dr. Porter coming to save the day. And Dr. Porter was able to take these data using the first time I'd ever heard of the word creed. I had no idea what that meant. And he put it into a grass suet model for Myrtle's Inlet and North Inlet. Let's look at Merle's Inlet. So the highest abundance of grass shrimp in Merle's Inlet was 235 grass shrimp per 25 meter stretch of stream. And you'll notice that's this little gray area. But notice these little white areas here. These are grass shrimp deserts. Marina, marina, marina. And then this is the jetties where the water falls in and they can't swim. It just washes them out in the sea if they're caught in that area. So we have these places that grass shrimp just can't live. North Inlet, let's take a look. The lowest cohort of abundance is this light gray area, which was the maximum abundance in Merle's Inlet. And that only occurs in the headwaters of these streams, and it's only 7% of the area. 93% of North Inlet has statistically higher abundance. This is the first time we were actually able to look at spatial data. And so, oops, let me go back. So what these data told us was that the grass shrimp abundances that I showed you earlier, let's go back. Those were reduced by about 90%. Those 6 stations told us that. Everybody said, Geoff, that's not enough. Geoff and Dwayne, that's not enough stations. So we went to 30. That was enough. And they gave us the same answer. So I only tell you that because without a place like North Inlet, we could have never explained any of it. And that's why these treasures left behind, like by Bill Baruch, are so important. We've been able to work with Dr. Kilpatrick and his research to show these PAHs not only affect grass shrimp, they also affect people. And we were looking at the port of Charleston thinking that we were going to have real high levels of PAHs right in downtown Charleston, where all the ships stop. But guess what? We found out that wasn't the case. When Daniel ran the data, the hot spot for PAHs is at the intersection of I-26 and I-95. Not something we had planned on, Dan, right? But Dan, always tells students, you get in the valley of the shadow of death when the hypothesis doesn't work out. So it's not the hypothesis that you pretty well know the answer, maybe. It's when you don't know the answer, what do you do? And Dan worked his way right out of that valley of shadow of death by clearly showing us that these effects were related to chronic obstructive pulmonary disease, and there were significant correlations with the PAHs and COPD deaths in those areas. First time anybody's ever shown that, I believe, in South Carolina. So we know first-hand cigarette smoke will cause cancer. Second-hand smoke, if you live with a smoke, that causes cancer. This evidence now shows third-hand cigarette smoke, i.e. exhaust from cars and combustion of fossil fuels, may also contribute to death. And this is really important information. This is where the debate is going on, and we're having this debate right now politically in this country. Do we continue to invest in fossil fuels, or are we going to go away from that because they may be adversely affecting all of our health? And they're going to continue to add to global climate change and other issues. Okay. Now, let's shift away from urbanization. Let's talk a little bit about climate change. So I want to talk a little bit about Vibrios again. These are some global trends, again, showing you population distributions. This is where all the people live. And basically, where people live, we have to treat their sewage, right? And we talked about this in class the other day. I asked you, what were some hot spots? Remember, we heard that in the lecture on global health from Dr. Anderson. I mentioned sewage treatment plants are hot spots. And you have to have waste treated. This is a study by Jody Harwood and one of her students in Miami And what her study, this is really profound. She shows a 1% increase in sewage discharge. The concentration leads to a 3-fold increase in Vibrio abundance. Wow. Everybody that's seen this paper, which kind of snuck into the press, suddenly said, wait a minute, are less people moving to the coast or more people? More people. More wastewater. More wastewater, more Vibrio. And we've already shown you that climate change by itself can cause a problem. Now we're once again back to that earth connection with urbanization. Dr. Sean Norman and his students showed that when we add sewage, it changes the gene expression in the Vibrios to select basically at 50% sewage effluent and 100% effluent. We upregulate genes that lead to increased virulence of Vibrio bacteria. That will make you sicker. So if we add more of them, longer periods of time, greater space, we've got a little issue to think about. Okay. I'm going to go through all of them. These are just showing you the data. So, evidence for that. 400, about 400 isolates, Charleston, to Hilton did, or excuse me, to Brunswick. And this is the number of antibiotics each isolate is resistant to. On average, the average Vibrio is resistant to 8 of the frontline antibiotics they can treat you with. Some isolates are resistant to all 13. So what are you going to do if it's resistant to something that we don't have an antibiotic for? Well, they'll have a new one. No, there's no drug companies in the U.S. developing any new antibiotics as of the last 5 years because they don't make money. So this is a global health issue. And so notice less than 1% of the Vibrios have no antibiotic resistance. This is clear evidence. Increased temperature, drives up antibiotic resistance. Increased levels of trace metals with urbanization drives up antibiotic resistance. All of this leads to highly antibiotic resistant bacteria. So one of the things we tried to do to control the runoff from urban areas is put in stormwater ponds. And in fact, we now, in 2008, we had 7,000. 2014, we had around 15,000. As of 2020, we had right at 21,000 ponds. These ponds collect nitrogen and phosphorus, and trace metal, and PAHs. And basically, the idea is we put all that in there and we won't have impacts in our estuary. But what if you live in one of those houses on there? And here's why it's such a big deal to do. This is a study by Diane Greenfield and Paul Sanderson, another study that we looked at. And what they show, these are looking at stormwater ponds, 2 stormwater ponds at Kiowa Island. These are when we have harmful algal blooms, these vertical lines. And then after those, we get bursts of Vibrio abundance in the ponds. So there's this intimate connection between stormwater ponds, the nitrogen and phosphorus in those ponds to drive up harmful algal blooms, which can be aerosolized toxins that can go right into your air conditioner. In Florida, they're actually now having to put HEPA filters on their air conditioners in certain places. And that's expensive because it can go right into your house. And so we now know there's this intimate association between harmful algal blooms and Vibrios. And that's what I read when I talk about climate change interaction. These are the kind of interactions we're looking at. Now, let's go back in time. The tabs we're working with are the cyanobacteria. They evolved some 3 billion years ago. What was the earth like then? It was much hotter and had a lot of what in it. CO2. Not much ice. So are we seeing a little more CO2 in the atmosphere today, a little higher? Yep, and we're seeing more of these. The dinoflagellates and dinothatons were all marine crab species. And they evolved about 4 million years ago after we evolved up. So you can really see the distinction between the marine head and the freshwater head. So this is the distribution. They cause fish kills. 40 to 50% of all the marine mammal strandings are caused by harmful algal blooms and closed beaches. This is the satellite image of Lake Erie. This is Toledo, Ohio. That's what the drinking water looked like. Their impact, 2014. So we're seeing these problems, and I'm not going to go through all the different species, but notice all these illnesses have one word: shellfish. Right. What does shellfish eat? Algae. They are a concentrator of these toxins, and that's why they're a big issue in terms of seafood safety. So, are these blooms getting worse? Well, here's a study from Puget Sound that shows the species Alexandra campanella. Presently, it's a problem 109 days a year when it's hot. But if temperatures go up 2 degrees C, it's now going to be 163 days out of the year, half the year. And if temperatures go up 4 degrees C, you're going to be almost a year-round problem when you can't harvest shellfish from Puget Sound. Why is that important? That's the second largest source of shellfish production in the United States. So this is a huge issue in terms of climate change. Our head's getting worse. This is PSP, paralytic shellfish poisoning, 1970, 2015. You tell me some of that's better monitoring, more monitoring, but everybody agrees it's related to climate change. Greater distances to the poles. Look in the southern hemisphere. We're seeing blooms there. We haven't seen them before. This is an international database. What country has the most blooms? France. Must be the wine. But the U.S. is close behind. Must be the beer. Not sure. But anyway, that's what the data tells us. And most of the events involve shooting. There can be foam events and airborne effects, and there can be fish kills and dolphin stranding. Most of these are these shellfish poisons. You'll notice the sign of bacteria now starting to crop up. Now, these are acute illnesses. These are not chronic illnesses. And when we talk about these cyanobacteria, we're talking about future chronic illness. And nobody's even tracking it very well because we don't have good data to know what some of the chronic effects are. So here's why you might want to pay attention to these freshwater events. One species, microcystin, accounts for 82% of all harmful algal blooms reported in the U.S., and that's a freshwater species. That was the species in Toledo, Ohio, in Lake Erie. Cylindrospermopsid and anatoxin, you add all those in, 96% of all hab blooms are in fresh water. They're not in marine. But all the illnesses are acute illnesses are in seafood. Why? Because the shellfish concentrate. The EPA lake study found that 39% of all lakes were detected with microcystin. So I'm not going to go through all that. Bottom line, we've been monitoring it. It goes up. In 2015, Brian Brooks and myself and a couple of colleagues, EPA, Jim Latour, Office of Water, we wrote this editorial. Are these freshwater blooms becoming the greatest public health threat in the United States in inland waters? And the next year, John Ravencroft from EPA published these data. We saw all of these are not blooms. These are actually health advisories where they had to close waters and shut down contact recreation and drinking water throughout the United States, mainly during the warm months of the year. These are the average concentrations. That's the average microcystin level in a bloom. That's the median. contact recreational water standard is 8 parts per billion. So if there's a balloon, stay in the room. Sorry. Okay, so I want to talk about a little lake outside of Columbia called Lake Erie, downtown of Charlotte. Second fastest growing area to the coast, population doubling time about 70 years, the coast is about 50 to 40. Very rapid growth. And these are some of the critters we find, particularly this one, Lindia qualia. It produces a toxin called saxitoxin. What do y'all know about saxitoxin? Probably not much, but I know in my classroom. The CIA used this to develop tips on umbrellas to assassinate people. Gary Powers, the U-2 pilot that crashed, and he shot in. He didn't have cyanide in his pocket. He had a pill with saxitoxin. So these are, this is probably the 6th or 8th most popular chemical known to man. This is what the shoreline of that lake looks like. This is Lindia qualia. It covers 2/3 of the shoreline of the lake. And what we have found, what drives blooms is the red field ratio. This is the ratio of carbon, nitrogen, and phosphorus. Remember those 3 cycles we talked about? If you disrupt your heart, your lungs, and your kidneys, you're in trouble. Same thing is true in the environment. You disrupt these biogeochemical cycles and affect the red field ratio, this is what you get. The normal ratio is 106:16 to 1. Anything greater than 20 to 1 is phosphorus limited. Nitrogen to phosphorus ratio, 10 to 1 is nitrogen limited. Those all put stresses that create conditions to allow this amount of bacteria to bloom. They can particularly thrive where there's no nitrogen because they can fix out of the air. Remember, they lived billions of years ago and they have an adaptation that allows them to do this. Other toxic algae can't do that. So these are the levels in lake watery, nitrogen limited, too much phosphorus. Recent paper published in the literature, the majority of phosphorus in urban areas is coming from what? Most of the nitrogen, I'll give you a hint, is coming from fertilizer. Where's most of the phosphorus from? Pets. Here we are, once again. Same old story, PAH, with a different verse. Kind of interesting. So these are the levels coming into lake watery, and these are the levels that leave. So that difference is how much is being deposited into the lake bottom. And so if we do this, we can predict that's the level of increase in phosphorus since 2002. In 2008, the population in Charlotte was 540,000. Today, it's over 970,000 people. And look what happened. Starting in 2012, we're going to get down here. And now we see it. How do you go swimming off your dock? You can't. How do you get your boat up? You can't. These are the distributions of the algae. In July, in green, and in red, it spreads throughout the entire lake, covering 2/3 of the shoreline of the lake. This is the drinking water intake right here, that little red dot from we got algae. That's the drinking water intake for animals. So, according to Duke Energy, 53% of all the mostly algae is Lindia. That's the big rule in the room. And so, Dr. Ferry and our chemistry group and Tim Shaw have been monitoring the phosphorus. And we tried to estimate with models, what if it's just nitrogen, chlorophyll, water column? The only thing that comes close to the amount of algal biomass, this is predicting algal biomass with nutrient level. The only thing that comes within the ballpark is phosphorus. So we clearly believe it's the phosphorus that's driving this. Now this is what saxitoxin looks like. It also produces the 6 other Lindia toxins. And they're present throughout the year. All these are present throughout the lake, throughout the year, in parts per billion levels. in the algae. Fortunately, it stays in the algae. It's an endotoxin. But if you dry the algae out, which they're doing right now because they've drawn the lake down, it releases the toxin. And so you can see up to 35 parts per billion, all the way up to about 45 parts per billion for the different Lingia toxins. We have conducted toxicity tests in the field. These are fathead minnows, some of Maggie Carson's. Those are our cages. And our results, no mortality. So that algae stays contained in the algae itself. But let me kind of make a point to you. If you compared the toxicity of this to mercury or lead, it's at least 100,000, a million times more toxic. If that's stored in a barrel at a Superfund site, you put a fence around it and don't let you go near it. What do we let people in the lake with? As I tell people at EPA, y'all are either way over-regulating Superfund sites or way under-regulating lakes, and maybe a little bit of both. But we clearly have to worry about the fact these toxins are present. There's 25,000 tons of that toxin in the lake through that cement. That's not a trivial amount. Our lab toxicity tests with the dried algae and the freeze-dried algae, these the tests we're running in the lab diagram. Again, live Lingia, no toxicity, live Lingia in the lab, no. Freeze-dried and air-dried algae, yes, it will be licensed still. Why do I say licensed still? They want to treat this with copper sulfate. That licensed still releases the toxin. Probably not a good idea. So we're debating that issue right now. Do we treat or not treat? And these are the levels, 15 to 20 parts per billion normalized algae. And here's your toxicity test result. As you can see, up to about 30 to 40% mortality. Now, our colleagues at Baylor, Ron Brooks, Geoff Scott, real modeler, he has predicted he can take the levels of nitrogen, the ratio of nitrogen, and if we increase the nitrogen phosphorus ratio, as we increase that, we increase the toxin production per cell of algae. So we now have developed, most of your forecasts for HABs, they predict the abundance of cells, but they do not predict the amount of toxin. And this is the first forecast that actually predicts the toxin themselves, so we can actually warn the public. But this is what makes you sick. So real quickly, I'm going to close on what about drinking water, since I mentioned those sites. This is Dr. Susan Richardson and her team at USC. They've been looking at these disinfection byproducts, and do these really potentially, we chlorinate this algae in the water, does that pose a hazard? And I'm going to go a little quick because I'm running out of time. So real quickly, I'm going to skip all the compounds that are formed and get to the chase. So when we basically look in South Carolina waters, they're highly enriched in iodine and bromide. In fact, Channel 10 in Columbia, the call letters are WIS. What does that stand for? Wonderful iodide state. That was our motto in the 1930s to attract tourists, because we had lots of iodide. That increased level of iodide, you can see here's your chloride, bromide, iodide, and that increases both the cytotoxicity by fivefold, and increases the carcinogenicity. This is some work by Mike Fleewood with Susan. These are some of the compounds we form when we have the toxic algae. They all contain nitrogen. Why? Because the nitrogen cycle is messed up. That's why. And so what do? The trihalomethane and fluorine they're not very carcinogenic, and we regulate them. But if we add these other compounds, they are much more toxic and carcinogenic. These haloacetonitriles, the haloacetamides, and halonitromethanes. And the big thing, there's no water quality standard for these in drinking water. They can put it in your drinking water. Nobody's looking. And what do we see when we look at watersheds throughout the state? With these algae, we chlorinate them. These are the chemicals that are formed. And the levels basically are double for the disinfectant chlorinated. Disinfection byproducts are double. And the genotoxicity is double when we have algae present in the drinking water. And that is a big concern because nobody is looking. So epidemiologists, I'm looking for some help. So I'm going to have to skip this. This is some of our human health work. Only thing I'm going to show you in China where they have taken blood samples. They have adjusted odd ratios of about 3, showing the correlation of hepatocellular carcinoma with microcystin inside people that are drinking water with these in the water. And if you drink alcohol, the synergism index is 4. So that's like pulling gasoline, because all of them damage your liver. This is a study we did in those humanized mice. And when we add Vibrio exposure following microcystin exposure, this is what leads to those severe Vibrio infections, that combination. So this is also an endocrine disruptor. I'm sorry, I don't have much time to talk about this. I want to just close on microplastics. The majority of bacteria growing on microplastics, food, our Vibrio friends once again, and some of the cyanobacteria. And in fact, Dr. John Weinstein has shown the average oyster in South Carolina has about 15 particles per oyster, mostly tyreware particles, which have been shown in Puget Sound to be carcinogenic in rainbow trout. And basically, John has found we can depurate these fibers out, but if you have a serving of oysters, you're going to get this combination of about 150 textile fibers, 54 tyreware particles. And so this is what those tyreware particles look like. So right now our research is really, our next exposure in mice will be microcystin, microplastics, and Vibrios. Because the Vibrios go on the plastics. The average abundance of Vibrios according to Ryan's dissertation, one particle has 1.25 million Vibrios and there are 15 particles. That means there's 22 million Vibrios in those oysters. The safe level is 1.25. These particles are going to cause major problems with safe consumption, and nobody is monitoring the microplastics. So, in closing, I hope I have shown you quickly that urbanization and climate change are here to stay. And they interact, and it's these interactions with climate change that are most profound. And in closing, let me just say, this is what Dr. Porter does, translating all this complex science to the public, is what we have to do, and nobody does it better than Dr. Porter and his team. And in fact, this was our first workshop here 5 years ago, right out here in the dock, meeting with all the folks from all the centers. And Glenn and them have developed the E.J. Strong and Environmental Justice Strong project. And they're using category approaches to broaden the scope and refine things. So I throw this slide up at the end. In 1854, the problem was urbanization. In 2023, it's urbanization and climate change. There's one common denominator, Vibrio.